So hello, my name is Andrew McNaughton. I'm a computational scientist in the modeling and simulation group in the computational modeling and simulation team in the computational biology group. Today, I'm just going to give you a sneak peek into a workflow that we're developing for functional prediction of protein structures with unknown function. So in the world of genome scale models, there's a large quantity of proteins that are genes that are known, have known function. There's also a large quantity of hypothetical proteins, hypothetical genes, and uncharacterized proteins. So our objective with this is to use AlphaFold and RosettaFold in order to predict structures for those and from uncharacterized protein sequences. And through that, using a suite of tools and using computational methods to see if we can narrow down a wide range of functions to a much more selective view so that we can pass them back to experimentalists to see if they're worth characterizing or if they play essential roles within metabolic networks. So there's two spots here that we accelerate our protein function prediction with artificial intelligence. The first is our structure prediction off the bat. So in our case, we use things like AlphaFold 2 or RosettaFold for our first step for artificial intelligence acceleration. And then the next step is we use that protein sequence, protein structure, and use another machine learning tool called P2Rank to figure out where along the protein structure that there are likely candidates for binding locations, so either binding pockets or surface binding residues. And from there, we can use not only the structure to compare to a large collection of other structures using a comparative database known as DALI. And this allows us to find structures that are similar to the predicted structures we have made. And there's a large correlation between structure and function in proteins so that we can see if we can, right off the bat, determine a large, narrow down the whole field through that initial prediction. But we can also collect things like if it's a catalytic functional protein, we can collect likely ligands and or substrates, and we can collect a large library of these substrates and run docking experiments on these pockets that we've predicted. And through that, we can collect a large quantity of evidence that may elucidate some of the functional capabilities of these proteins, and we can make predictions based on all of the previous steps of the process, and potentially enhance the annotation in order to give experimentalists an idea of their role in a pathway or the role in the cell. And you can also do this for not only these catalytic proteins with protein ligand interactions, but you can also do the same thing for protein-protein interactions. If it's a protein that would provide allosteric benefits to other proteins, that's a potential application. In the end, we would want this to be a end-to-end sort of automated workflow where we just pass it along we strip out a genome of all of the sequences that are uncharacterized or unknown or hypothetical, pass it through a pipeline, and we can get all the way down to a set of predictions that we can pass back. So that I have a poster outside. So if you have any specific questions about either what tools we use for any of these pieces, you can come out and ask me. There's also a little bit more detailed breakdown of the workflow.
Thank you very much, Andrew. Our next speaker is Garry Buchko, and he's a senior researcher here at PNNL, specializing in structural biology using NMR. Welcome him.
Late afternoon, everyone. My name is Garry Buchko. I'm a NMR spectroscopist / structural biologist here at PNNL. And what I would like to do, first of all, is thank the organizing committee here for this opportunity to present this flash talk, because I think what we accomplished here is really something that's very, very flashy. And that is, we've been able to express in the bacterial system a phosphorylated protein, a protein that's phosphorylated on a specific site in the primary amino acid sequence of the protein. And you know, I only had so much space for the title, and I'm missing a couple of adjectives for this accomplishment. And one of them is this phosphorylation is in high yields. We're able to obtain like milligrams, over 10 milligrams of protein per liter when we do this. And also, this system is also very fidel. We have incorporation of phosphoserine that's greater than 90% in our protein. So another things that's important about this accomplishment is that it is also the first example where we have a phosphorylated protein that's also been triply labeled, because as an NMR spectroscopist, sorry, I don't know if I'm doing this, or I guess I am. Okay, as an NMR spectroscopist, you want to label your protein as well with NMR friendly isotopes. Now, this was possible with collaboration with Ryan Mell and Rick Cooley at Oregon State University. And over here, I've shown the Oregon State University Beavers logo. Now, I put this logo up here not to please Scott Baker, nor because it is the emblem of Canada, but because beavers are known for their great-looking teeth. You know, on the surface of this teeth is a biomineral. It's enamel. And it's one of the hardest tissues. It is the hardest tissue in invertebrates. And it's actually got a very simple chemical formula. It's just calcium and phosphate that crystallized from hydroxyapatite. But this hydroxyapatite is being allowed to crystallize by this protein amelogenin in long, thin strips. And more importantly, amelogenin weaves these strips into a form that makes it very, very tough. And it's you know, for beavers, it's not so important to have, you know, teeth you know, it's important to have enamel that's good for your teeth. But with beavers, their teeth are growing all the time, and you actually have to keep on chewing wood so that the teeth don't grow into their bottom jaw. But as you know, for humans, after we lose our teeth, they're gone. There's no way to repair these teeth. So it's very important to understand how this protein is forming enamel, because we could put a lot of dentists out of work if we're able to repair teeth or even go get the teeth to come back into place. Now, the system we've done it is that because the people at Oregon State University, Ryan and Mel, have a genetic code expansion laboratory set up. And on paper, there's various different flavors of genetic code expansion. But on paper, what they simply do is use the Amber codon and create a transfer RNA that is specific for the amino acid that you want to introduce into your sequence. And I say on paper, it looks pretty simple, but in practice, it has been very hard. And very recently, Mel and company were successful by doing a lot of genetic modifications to the host that's expressing these proteins. And in the third generation of this protocol, they're able, we finally having success. So what they've done is they just took the amino acid, the genetic sequence for amelogenin. Or what I might also point out, though, I forgot to say that this is the primary amino acid sequence of the protein. There's 16 serines in it, but only one of them is phosphorylated, the serine at the 16th position. And there's evidence that this phosphorylation is very important for its biological properties. And a group here at PNL that I've been working with for the past 10 years, and people all over the world are largely using non-phosphorylated amelogenin for their biophysical chemical studies. So it's important to get this protein with the with this modification introduced here. So anyways, making a long story short, Mel and company sent me a plasmid that contained the construct for the transfer RNA, and also a construct to express the protein of interest with just one change, putting a stop cone in the middle. And right off the bat, first time I tried it, I took the sample. I didn't go to I took the sample because NMR phosphorus is NMR friendly, collected a 1D spectra, and lo and behold, there was one signal for the single phosphoserine in the protein spectra. And over here is HSQC spectra of phosphorylated and not phosphorylated amelogenin. And you can't probably see it here, but the peak for serine 16 disappears in the not phosphorylated form, but it appears in a new position in the phosphorylated form. And of course, here we have a gel, and this is a phosphogel. When you run a phosphoprotein in the gel, it retards its migration on the gel, and you can see bands for the phosphorylated protein and the non-phosphorylated protein. So this is very important because phosphorylation is the most important post-translational modification to proteins that in nature. So now we have a system where we can make label proteins for further biochemical studies. And that is all. Thank you.
Okay. Okay, our next speaker is Minjae Kim. She's from Oak Ridge National Lab. So please welcome Minjae.
I cannot share my screen right now. It says you cannot start screen share while the other participant is sharing.
Yeah, we are changing the settings.
Okay, let me try
Now
this
you
time.
should be able to do.
Okay. Can you see my screen?
Yeah, we see it now. Yes.
Perfect.
Yes.
And
And we
thanks.
hear you very well.
Okay. Thanks for the invitation. And I'm Minjae Kim from Colorado State University. And today I'm going to talk about the human microbiome study that I did recently. So the infant gut microbiome has lifelong implications on health and immunity. However, there is still limited understanding of the microbiome differences and similarities between children in low and middle income countries versus high income countries. So we described and compared the microbiome profile of children aged under 48 months in two urban areas, which are Maputo, Mozambique, and Atlanta, USA. First result is alpha diversity. So here you can see the non-pareil diversity. This represents the combined richness and evenness, and showed that the gut microbiome of Atlanta children was more diverse, especially in 12 to 23-month-old group and 24 to 48-month-old group compared to the Maputo children. These results might be associated with growth faltering in sub-Saharan African countries. And interestingly, this is contrast to the previous studies result based on the 16 assembly sequencing data conducted in the Malawian children gut microbiome. So specifically, they showed that the Malawian children, the older than 3 years old, showed the higher diversity compared to the age-matched US children, and no significant differences in alpha diversity under the 3 years old age groups. And secondly, we tried to assess the relative abundance of the specific pathotype of the E. coli by using the diagnostic genes abundances. And before moving on, in the x-axis, you can see some of the legends. Says A_ATL, A_Maputo. So A means the youngest age group, 0 to 11-month-old, and B is the second, and C is the oldest one. And then ATL is Atlanta, and Maputo is Mozambican children. The results suggested that we have a higher relative abundance of specific diagnostic genes for several pathotype E. coli, such as enterotoxigenic and enteropathogenic E. coli in Maputo children's gut microbiome compared to the Atlanta one. Next result is related to the antibiotic resistant genes, and this is the global health problem, and high prevalence of antibiotic resistant genes in the gut might be related to the presence or outbreaks of antibiotic resistance pathogen. We use a tool called Deep ARG that can predict ARGs from short metagenomic reads based on the deep learning model. And results here suggested that we have a higher relative abundance of total ARGs in Maputo children's gut microbiome, especially in 12 to 23-month and 24 to 48-month compared to the age-matched Atlanta children. And this is primarily driven by E. coli, as shown here at the heat map, and the other species. So we deeply look into the E. coli metagenome assembled genome, and in the last heat map, left panel indicate the Atlanta E. coli max. So the rows indicate each of the different antibiotic resistant genes, and columns indicate each of the E. coli MAC. And the right panel indicate the Maputo E. coli MAC. So as you can see, there are more and diverse ARGs found in the E. coli MACs in Maputo children's gut microbiome, and also the blue dot indicate the co-occurrence of those ARGs with the mobile element on the same contig, and this suggested that potential for the mobilization of clinically relevant ARGs in E. coli. Collectively, our research revealed that pathogen load and asymptomatic infections in Maputo are highly prevalent and require action toward developing a healthier gut microbiome, which may support long-term health and well-being.